Good evening, everybody. It's great pleasure to have Claire Wyart here today with us. Claire did her undergraduate. Excuse my French pronunciation here. I'm going to try. Claire did her undergraduate from École Normale Supérieure and PhD in biophysics from the University Louis Pasteur. And then actually I learned just today that she spent a year in Nepal and India teaching science to Tibetan school students, for which she's even won awards. And then after this, she did postdoctoral fellowships with Ehud Isakov, where she started to use zebrafish larvae as model organisms to understand spinal circuits. So in 2011, she started her own lab at the Brain and Spine Institute in Paris, which is where she is today. And her lab studies spinal sensory signaling. Her research has been funded by the European Research Council, the HFSP, and recently she was also named the Robertson Investigator by the NY Stem Cell Foundation. She has won many awards for her work, including the EMBO Young Investigator Award, member of EMBO, and the Fence Kavli Scholar, and the French National Order of Merit, etc. So it's a great pleasure to have you here, Claire. Thank you very much for accepting our invitation, and look forward to hearing your lecture.
Thank you very much, Pat. It's really nice to be here. And I really want to, as much as we can on Zoom, try to make it interactive. So please do ask questions and do interrupt. That would be very nice. What we're going to do together is discuss about the control of movement. And I'm going to show you the effort that we've pushed in one direction over the last decade already to try to dissect the circuits in animals that are transparent. And so you can actually dive without, it being invasive into the brain and spinal cord, just using light in order to remote control by optogenetics and also monitor the activity of cells in vivo. And the way we're going to do it is that I'll start today with my presentation focused on the role of sensory feedback and understanding how the kinematics are shaped during movement. And tomorrow you will have a chance to have the presentation from two people I work with. One, Gautam Reddy, who is a theorist who has been developing a new method for segmenting behavior that is, I think, very useful for other animal models. So I'm really excited to share that with you. And then Gautam Sridhar, who has been using the method developed by Gautam Reddy in order to look at sequences in the behavior. So I see that Vatsala put, raise your hand if you have a question or post it here. And so feel free to raise your hands and if you can, make it interactive so I can also hear you, that would be super nice. So I'll start by just going really into the basics, but I think it's always good to be reminded the essentials when you start. So, what is really remarkable about movement is the fact that we don't really think about it when we do most of the movement that we're doing during the day. And in fact, it's also a reason why we still don't quite understand it, because despite being so essential for survival, any of our behavior require movement to occur. We don't really consciously think about everything we do in order to move. And that's somehow a little bit of a limitation because, as it turned out, for many human things and things that request our creativity, what we think we do consciously, we tend to pay more attention to. And so in the field of neuroscience, even though the first evidence for synapse between neurons was found in the spinal cord, in the motor circuits by Eccles, looking at sensory and motor integration, we are still in a stage now where, surprisingly, if you have an accident and you can't move because you get a surgery, spinal cord injury, there is not that much that we can do. And if the communication between the spinal cord and the brain is disrupted, really, there is not that much treatment that we can implement. So how do we capture movement and how do we quantify movement? So the way that people have looked into this, in particular, this person, Étienne Jules Marais, who is a professor in Collège de France already a century ago, he had the obsession of understanding movements, and he got the idea that it was possible to capture movement by doing something very simple that nobody had done before, which was to take images that you superimpose of the movement while it's happening. And so he developed this device that was called an imaging shotgun, where he could take images of the movements and ask people to wear often white clothes in order to appear white on a black background. And he could take images at 12 times per second in order to really capture all the kinematics of movement in those instances where it's too fast to be perceived even by the human eye. And so he did that on many, many, many different kind of movements. And then from those superimposed images, then he drew the skeleton of the limbs and the body, the head, so that he could retrace the movement and quantify, put numbers on it, so that even though the movement fades away, he can have a trace of what happened during the movement. And this he called the chronophotography. And I think one important and interesting point for the history of science is that his effort, because he was so obsessed about movement, actually led to the cinema and to the cinema that we know today, because his idea of doing chronophotography, he gave it to the Lumière brothers, who realized that they could do something, they could create a new path for art and not only science with this tool. And so now if we try to go back to biology and neuroscience, what do we understand of the control of movement? So this is taken from a review of Olekin a few years ago. In the mammalian, brain represented here, but it's true for really, most of it is true for all vertebrate species. You actually have a control of voluntary movement that relies on structure in the brain, where you have basal ganglia here that provide very important inputs to a region called the mesencephalic locomotor region that is necessary to initiate locomotion. And in Parkinson patients, the disruption of this pathway leads to a lack of initiation of movement. And then from there, in this region in the center of the schematic here, there are projections into the hindbrain, into the reticular formation that projects down to the spinal cord. And these are called command neurons. They are in the brainstem. And they really send the information to start and initiate the beginning of a locomotor bout. All species move as bouts. And so in the spinal cord, you have circuits that are called central pattern generator. They are like little oscillators that will be really critical to send and produce a movement by sending the output to the muscle via motor neurons that send very long axon out of the spinal cord. In humans, in particular, those motor neurons need to go from the lumbar region in our spinal cord all the way down to the tip of our toes. So they are like typically 1 meter long. And then from there in the muscle, you have different systems for sensing the contraction of the muscle and also the tension in the Golgi tendon. And this will be sensed by sensory cells that then project back to spinal cord in order to modulate the activity of the oscillator in the spinal cord. So one remarkable experiment that was done by Graham Brown, again, I'm using old reference here, but I think it's nice to think about those people who started the field. Graham Brown did an experiment where he put a decerebrated cat on a rolling mat, as you can see here. And he, at this time, was very low key. So he changed the speed of the rolling mat by hand. And what he observed, so when you see, even though the movie is low quality, you have like cable that hold the weight of the cat, because the cat is able to, induce locomotion based on the sensory stimulation, but not carry its weight. So he has to carry the weight. And what he observed is that at low speed, the treadmill leads to initiation of locomotion that looks like walking. And then when he increases speed, he can see that it changed the gait. And just from the sensory feedback of these rolling mats, is able to actually get the animal to trot and to gallop. So that's really interesting because it shows us that most likely this feedback is sufficient, in fact, to induce those change of gait. But how can we understand the role of feedback in the system while it's moving? And so one of the problems that we've encountered is the fact that the locomotor oscillators that I talked to you about, the central pattern generator, they're confined to the spinal cord. They're very, very much difficult to access, typically in humans or in the cat, because they are inside the spine, right? So it's very intrusive and difficult to record from them. And then in order to understand what the sensory feedback does, you would need to ideally record from these neurons while the animal is moving. So this is adding another layer of complexity because you actually need to be able to monitor the activity of cells while the animal is moving in a structure, the spinal cord, that is difficult to access and that is moving too when the animal is moving. So it makes it really tricky to do. And so what people have done in order to understand the organization of the motor circuit is to look at a simpler animal than the cat. And a lot of our understanding comes from this animal that you see on the left. That is a parasite called the lamprey that latches on fish, as seen here. And this preparation, the lamprey, has been very useful because you can dissect the preparation. And it was a work of Ramanathan in the 80s. And you can put the spinal cord in a dish. And when you're recording from neurons in that dish, you can keep them for very long. So the preparation can stay for a very long time in this dissected form. And then the nice aspect is that you can elicit what people have called fictive locomotion, because of course in this current setting the animal is not actively moving, but what you can do is by adding neurotransmitter like glutamate or neuromodulators like dopamine or serotonin, you can elicit an activity pattern that looks very much like movement. And it has in particular this alternation of left and right activity. When the animal moves, the lamprey forms ondulation of the body that propels it forward. And these ondulations are going from head to tail, typically during forward motion. And they are occurring with contraction of muscle alternating left and right side. That's what you can see here on what is called the ventral nerve root recording, where now an electrode is brought next to the motor neuron bundles that go outside of the cord, and you can see the activity of many of those motor neurons activated on the right or on the left, right and left. And this is the activity pattern that will enable to sustain the contraction of the muscle in order for the animal to form those ondulation of the body. And so based on multiple decades of work from the team of Stan Greener and many others, it's been possible to establish that in the spinal cord, a very simple schematic of how the network is organized is depicted here, where you have on each side, the left and the right side, small networks that are constituted of excitatory cell together with inhibitory cells. And those are premotor interneurons that will project onto the motor neurons, M cell here, in order to send the command to the muscle, and the contraction occurs. And then the sensory feedback, as you can see here, they're called stretch receptor here. They can feedback and send information back to those central pattern generator. And in order to initiate the voluntary movement, you need a descending command from the brainstem in particular. And so the way to elicit locomotion is typically either via this command, you say, I want to walk, and then you start walking, or via reflexes that will involve the sensory cell in order to elicit the initial movement. But the remarkable fact that, didn't encourage to investigate too much the role of sensory feedback is that you don't really need the sensory feedback in order to get the oscillation, because in the fictive locomotion here, you don't have the sensory feedback. The muscle cells are either completely dissected out or they're not contracting, and you can see those oscillations very nicely organized. So just close the door so that you don't hear too much of the so this is like, sensory feedback is really not necessary for each oscillation to occur. And this has been a debate that was very intense, in fact, in the 80s. And this preparation, the fictive preparation, somehow the fictive locomotion in vitro kind of settled that the feedback was not necessary at each cycle for inducing another one. But it was still not clear, well, really what happens when you don't have the feedback, because people didn't really have the tool to look at this question. So what we know from the work that has been done by Tom Jessel with his collaborator is that the organization of the spinal circuit, and it's true also for the hindbrain, is remarkable in the sense that you have in all vertebrates the similar organization with domains that are written here with names, for the motor neuron and also for different kind of interneurons. And those domains are very conserved across species, so that you can look into a fish or a mouse and you find the markers that are the same to define the cell types. So by doing this effort, collaborators of Tom Jessel, Martin Goulding, and others have really established that it was a very nice way to investigate the organization of the circuits with molecular tools in order to now understand what is behind each cell type in the central pattern generator. So thank you, Vatsala, for answering the question I see online.
So any routine questions like that, I will take care of, but I think
okay,
more
good.
involved questions, I'll deflect to you.
Okay, good. So
I think Vijay has a question.
Okay.
Yeah, Claire, is the same circuits for both vertebrates and invertebrates?
So that's an important one. Yeah. So in fact, this is really true for vertebrate species. But in invertebrate species, we have evidence that similar organization occurs in the sense that you get indeed descending commands that are projecting onto circuits that control the activity locally of the motor neurons. But then it's very different in terms of transcription factor, we see them here. Each transcription factor defined here will, specify a progenitor domain that will lead to, typically from what we understand now, about at least 1,000 cell types in mammals. And so that's very different. So in fact, in invertebrates, you really have, the neurotransmitter are not the same. You reverse acetylcholine and glutamate compared to vertebrates, and the transcription factor are different. However, there are very similar rules about how the circuit seems to be organized. But it's still early days from what we know of Drosophila and C. elegans, but it's not exactly the same. It's not conserved in that respect. Yeah.
Thanks.
I see that Sanjay raised his hand.
Yeah, I had a question. Hi, Claire.
Hi.
So in the experiment with the cat, the decerebrate cat, the stretch receptors were part of what allowed. So there's some interaction between the treadmill and the stretch receptors were part of what generated rhythmicity.
Exactly. So in the cat, the stretch receptor are the muscle spindle that we also have, right? That are sensory cells that will form nerve ending on the muscle itself and will feel the tension. like the contraction of the muscle, and also the Golgi tendon with afferent that are on the tendon in order to know how much force you have on the tendon. And so
Right.
what is really cool, experiments in the cat are not that easy to do now, but what is really cool is that, by just relying on the feedback of the sensory pathway, the cat initiates locomotion and adjusts, its speed and gait to the speed of the treadmill.
Right. So this is sort of, what we removed there then was just the sort of descending locomotor command from the brain.
Exactly.
But everything else which is not how we think of CPGs today, right? We think of CPGs as essentially not as being, independent circuits within the nervous system that generate rhythmicity, even in absence of sensory feedback. I mean, maybe a little bit.
So they've been isolated by, as a matter of fact, in those fictive locomotion prep. But when you think about it, they're never isolated, right?
Yeah, correct.
So that's why, there are very interesting experiments in humans on patients who had tuberculosis, where it's only in Russian, and it's very hard to share this, but where you see that but to my knowledge, the only evidence in humans that we have also CPGs where they've done electrical stimulation on those patients, and you see the two legs doing that. But it's clear that we receive continuously information from the sensory system, right?
Right.
So of course, when you get, the Babinski reflex, when you touch your knee only, you just, you just get the reflex of, one bent.
The knee jerk reflex. Yeah.
Yeah. But then when you are indeed walking, or when you are yourself, exposed to more complex stimuli, you can develop, more interesting response. And so what is interesting is that this, this role of the sensory feedback has been a little bit put aside due to the fact that people were fighting to say that the oscillator were completely autonomous. And that's why it's somehow interesting to reconsider what could the feedback do. So when I started to ask that question in our lab, I realized that I was facing an audience of physiologists who were working on the central pattern generator for many years. In one of the important reference center on this topic, and I asked them, what do you think happens if you remove the feedback during locomotion? And what was funny is that none of them had an idea. Like, none of them dared to say something that could be stupid, probably. And they couldn't do the experiment because they had a preparation that was in vitro, completely isolated, and to some extent looking quite similar. So let me tell you how we've tackled this question now. So this is one review from 6 years ago that explained how, with the molecular tools, people identify different progenitor domain, and they find, those V2A cells expressing VSX2 or SHOX10. Then they define the V0V, V0D, and V2B, and so on. And they built, this is a work of Rybak, they built like model that try their best to explain how the cell types are organized in order to initiate locomotion. And you can see it's very complex, and on top of it, it's not, exhaustive, because we are missing a lot of neurons that we didn't record that much from, thinking about the V3, for instance. But it's true for other ones that have been not fully characterized. So there are clearly important cell types in the ventral spinal cord that control locomotion by modulating the activity of the motor neurons. And they are interestingly conserved across vertebrates. So we can look at swimming species, understand how they are organized, and then find out from a very simple animal that, just undulates the tail, how do we, evolve with different complex features when you need to control limbs and so on. And so that's been the motivation of the field. And so what I'll do today is to really show you how we've been looking at the kinematics of single locomotor bout those, discrete events that constitute locomotion, because we never locomote continuously. No animals does that. And then we'll focus on the sensory feedback, because it's a nice way to show you the different tools we can use to experimentally tackle this question when animals are freely swimming. And then the second step will be trying to take it to the next level by asking, with the work of the tail of the two Gautams you see here, asking, well, now if we try to look at locomotion during real life, when you're really doing something, can we actually segment in the locomotor pattern sequences of actions that are recurrent in the behavior, so that we can start thinking, okay, not only do we generate, a motor contraction leading to a single quantum event, but we can also look at it as a series of events. And even in animals that think that they are very sophisticated like us, there are many things we do that are semi-automatic, so that when you wake up in the morning, you probably have a routine and you don't even realize, but you actually come up maybe from the same side of the bed, maybe you sleep on the same side, and then maybe you walk and do your first steps completely without thinking. And that is also the case where if you're walking on something on the floor and you have to react immediately because you injure yourself, you have a response that will not be a simple reflex like the Babinski one on the knee. It will be a series of movements that would enable you to make sure that you're going to be safe, and you check what happened. It's the same for escaping a car or many other things that can happen to our life, even though they are not stereotyped. They are clearly more like integrative reflexes that involve also the brainstem and not only the spinal cord. So it's interesting to think about that and to try to see in a locomotion now and motor control coming from this, bottom-up experience, like going from the sensory feedback onto the brainstem. How do we understand the structure of movements, and how can we, dissect the mechanisms that, define the kinematics of movement, but also the long-term structure in movements. So I told you, the feedback comes from most of what has been studied, and it's still the case in the textbook. The feedback comes from outside the spinal cord with sensors that are on the skin or also on the muscle or on the tendon. And they are part of this dorsal root ganglia that, you see here one neuron with a T shape, one axon going all the way to the organs, the skin, the muscle, the tendon that are in the periphery, and then another axon going into the spinal cord and modulating the activity of interneurons and motor neurons in the spinal cord. And then the motor neuron goes back to the muscle fiber and enable the contraction. So this schematic is, from Charles Sherrington. It's also, one of the founder of the field. And it's interesting because, as I said, the question has not been tackled very much due to the difficulty to access the circuits in vivo during locomotion. So it's really hard with electrophysiology to ask this question because you don't get the mechanical stability, but what can we do with optical methods? And on that one, I'm going to check the question. Yes, so I see that it's more of a comment of Sanket saying that, another example is like driving a car, riding a cycle. You don't think about it. And in fact, if we go back to the Parkinson patient, what is really neat is that in Parkinson patients, and that happens to more and more people in the world, when you actually can't initiate movements, you're going to still be able to ride the bike. And so if you put Parkinson patients, you can find that on YouTube, and you push them on the bike, they won't be able to initiate their movement. But when you push them on the bike, they will just go and roll and, bike and continue biking. Similarly, there is also a really nice movie that you can find online where Parkinson patient who was a soccer player his whole life is unable to walk in the corridor with the nurse. And then the nurse have the idea to use a tiny little soccer ball that they attach to a string and they show it in front of the patient. And the patient suddenly sees the ball and he really wants to do what he's done, before all his life. And so he starts kicking. He starts kicking the little ball, and by kicking the ball, he can actually walk. And so there is this really nice interplay of sensory inputs and voluntary movement, where indeed, if you lose this drive from basal ganglia and dopaminergic input to the mesencephalic locomotor region, you can catch back by having this sensory input coming either from feeling that your foot are on the pedal and you're pushed to bike and you feel that you're on the bicycle and you can continue to do it, or I said for the ball. So now I would like to ask you guys, what do you think we could do to investigate this question with optical methods? What could we do with optics and light that we cannot do with an electrode? Just say anything really, like, you don't have to have the right answer. It's more for a discussion, because that's what we normally get for a school. A bit of a discussion. Yes, so non-invasive, Ms. Ali, you're perfectly right. But what do you think we could do? Exactly, like what do you think is possible? Yes?
Hi, Claire. Basically, what you can do is that you can have those implanted electrodes, and then you can shine light, and then you can track the animal and see how stimulating using optics and the behavioral response that can be like seen simultaneously.
Yes, so you can indeed activate neurons with light. That's one way. So you can say, okay, let me try, I'll activate the sensory cell and then I'll see what happens. That could be one way. But what if we try to understand also the role of sensory feedback, what else could you do you think?
One more thing that was coming to my mind, like that has been a recent advance, is this use of this dread receptors. So we could use DREADS technology to, like without these optical cables and all those things, like we can stimulate the particular brain region and then see the behavioral response.
Yeah, totally. So we can use light to avoid indeed the issue of the mechanical stability that you need to keep for electrophysiology. So we can use them, like you said, to manipulate the activity of neurons. That's one way. So you can do a big perturbation. That's what I've done a lot in my postdoc. And then you see what is the consequence in terms of behavior. And then another one is to try to record from them without having to put an electrodes and keep it stable in the spinal cord, because it's very hard to do without damaging the motor function itself, right? Sanjay, you have raised your hand too?
Oh, you have to slow my hand. Yeah, I was speaking. No.
Okay. So the model that we use to do this optical manipulation is like the larval zebrafish that you can see here. And in fact, it's a very, obvious reason that motivates us to use zebrafish, because as you can see, it's transparent at the larval stage. So what is really nice is just with a fluorescent microscope using visible laser, in this case, spinning disk microscope, we can image, you see all the neurons that are in the spinal cords, in the zoom region, and also in the brain. And it's a very nice feature, because we don't have to dissect the preparation, and we can also look at what happened in the brain or in the spinal cord while the animal is moving and beating the tail. It's a genetic model organism, meaning that we can use a lot of the new technologies like CRISPR-Cas and other approach to generate transgenic animal like this, where in this case we have a sensor for calcium that will give us a readout of the activity of the neuron in cell. And then we can also perform electrophysiology. Vatsala is an expert in that, and we can monitor the behavior. So it's relatively simple. It's another swimming animal, but it's fast. It goes from 15 to 100 hertz, and so you need to capture the movement at really high speed. And so we have different ways to either read the activity of neurons. We can use the sensor, like I mentioned, for calcium GCAMP that will change fluorescence with the calcium binding, increasing fluorescence of the GFP when the calcium binds to calmodulin and the peptide M13 here. And then we can also use opsins. So this is an example for chirodopsin, but there are many opsins now that will enable to activate or silence neurons during the behavior. Okay. And so now if we look at the behavior of the fish, they have very stereotypical behavior, starting already when they're hatching at 2 days, they are good at escaping from predator. And here in this case, they are really good at avoiding to be sucked up and eaten, you can see. And then at 5 days, they need to actually hunt for food. If they don't hunt for food, they will die of starvation. And so this is one example where you can see a little paramecium swimming in front of the fish in linear trajectory, and the fish will realign itself in order to eat the paramecium. So it's typically what we need to do, except for us, hunting is much less, challenging, but most of the time. But it's typically what we need to do, right? We have a motor sequence that enable us to capture, in this case, food. And the sequence needs to be reliable, it needs to be effective, and the animal needs to do it only at 5 days of age. So you will hear from Gautam tomorrow how, in this particular case, if we start to analyze each bout that the fish is doing, we can put them together and find out what is the sequence that an animal has used in order to catch a paramecium. And that enables you to actually dissect the behavior in more long-term dynamics. So in the beginning of the lab, we've been developing with Olivier Mira a tracking algorithm called ZebraZoom in order to look at multiple animals in a well here, and then we put many wells together so that we can look at hundreds or thousands of animals to conclude from. And the way we do the tracking is very similar to the other teams. We put an axis for the head using the 2 eyes and the swim bladder to recognize the direction of the head, and then we put those points along the tail so that we can look at the tail angle over time. And you can see when you monitor just one at a time that most of the time the larva actually stays quiet, it doesn't swim. And then about 10% of the time on average, it will start to initiate those bouts that are discrete. And then it swims spontaneously. And when it encounters another larva, it will react by having an escape where it reacts the same way that it did for a predator. Okay, so this tracking is very important because it's what enabled us to put numbers, and now the ZebraZoom software is available on open access, so you can directly use it if you're interested. So when we've done the tracking exactly like the same way that Etienne Jules Marais was doing his superimposed image, we can look at the superimposed image, we can look at the tail angle over time, and we can also compute the curvature, how much the tail is bending as a function of the position on the rostral to caudal axis. And what you can see here for the spontaneous slow forward swim is that the tail angle is of low amplitude, and it corresponds to contraction of the tail on the caudal end. So you see it's the second half of the tail that bends, and it's also what you can guess in those superimposed images. This is very different from what they do when they are escaping. So I showed you one movie of an escape, and now you can see the superimposed image. So during an escape, the animals start by this massive, bend of the body, that's called the C-bend, to go away from the stimulus and then swim really fast So this is the same duration of the slow forward swim. So it accomplished a much larger distance, and it does that by doing tail angles that are larger in amplitude and also occur at higher tail bit frequency. And the combination of power with large amplitude and the tail bit frequency at a high that is higher, that will lead to higher speed of locomotion. And what is remarkable in that behavior is that now the curvature, meaning the contraction of the muscle, occurs from head to tail during the C-band, and then gradually, transfer to the same or similar pattern that we've seen in the spontaneous slow forward swim, where you have a contraction of the tail occurring only in the second half here. This is distinct from the routine turn that the fish will do spontaneously to orient itself when it's just exploring an arena. In that case, head to tail contraction of the muscle, but very low tail bit frequency, so that you have over time very slow oscillations from left to right. You see this is lasting much longer. This is 50 milliseconds. So this is lasting much longer than a cycle here that occur typically at 80 hertz at the beginning of the escape and can go all the way to 100 hertz, meaning like 10 milliseconds between each band. Okay. So now let me see.
There's a question. I don't know if you already answered the question or not, but the question is, could you please explain the heat maps?
Yes.
Which you.
Yes. So so here is like the curvature of the right in red and left in blue. And so high value of the curvature will be either red like here or very dark blue. Neutral would be green. So you have 0 at the beginning before the movement starts. And then during an escape, you have the high value of curvature on one side that is followed by an oscillation, with high curvature on the other side. And then you go back and forth between each side. The maximum curvature is at the beginning, and then it goes, from head to tail here, and then progressively into the caudal end only, so that the slow swimming occurs only with contraction of the tail. This, I apologize, was not specified here. This is like 50 milliseconds, I believe. So you have typically a movement that we call about is occurring in the range of 200 milliseconds from beginning to end.
There's another question. Vijay has a question and has raised his hand.
Yes.
When you say the curvature, I presume you're thinking of this like a planar curve, and the fish itself doesn't have a torsion along its midline?
Exactly. So, well, actually, you can see a little bit here that there is a movement in the rolling that I will maybe go to, because we've looked at that also in terms of sensory feedback. But most of it, since we're imaging from a camera above the animal, we're looking at it from a 2D plane. And then what we're doing is that for each position, we're calculating the radius. So here, if you look at the C shape of the body, we're calculating a radius to find out, well, what would be the, at each position along the tail, what would be the, the radius typically that define the curvature. So if we were fitting a circle, what would be this radius? If the radius is very, very large, when the tail is straight, then you have a very low curvature. And if the radius is very small, then you get a very high curvature.
Right, thank you.
Okay, I saw another hand raised, but I'm not sure if it was solved.
Yeah, thanks.
It's okay. So these are typical type of bouts that we're seeing. And then if we refine the analysis, we can detail that there are multiple, there are subtypes of those slow forward swim or those routine turns based on the amplitude of the oscillation and the number of oscillation typically. During an escape, you see that you have a very nice relation between the amplitude of the tail bend and the frequency, so what we call the tail bit frequency in hertz here. So you see that the scale is inverted so that it's 100 hertz here and 14 there. Okay, so if we color code the first, the second, the third, the fourth, and the fifth band, what we see is that during the first bands, we have this very large, you see, tail band, that is the C band with very large value of the amplitude of the tail angle. Typically, you see between like 80 and 120 degrees. And then gradually, we go, along that line with high amplitude, high tail bit frequency, and then gradually the movement will evolve into lower amplitude of the movement and lower frequency. And so you can see this response a little bit as a dampened oscillator, where you start with a very large oscillation occurring at high frequency, and then gradually you reduce both amplitude and tail bit frequency together. And what a lot of work has shown, which I'm not going to review entirely, is the fact that if you look at the motor neurons, they are different kinds. The first to be born in the early stage of the embryo are the primary motor neurons. And these guys are only recruited for movement that occur above 30 hertz. So the fish during embryogenesis really built already right away the circuit that enable the escape so that it can survive when the egg hatch. And then with time, there is a second wave of motor neuron that will involve what we call secondary motor neurons, because they arrive in the second wave. And those motor neurons will carry the oscillation at the low frequency range under 30 hertz. So this is really interesting because it shows that, during the development of the system, the animal right away puts in place the escape in order to swim fast and away from an aversive stimulus to ensure its survival after hatching. And then it has to do what I've shown you, the hunting. Otherwise, it dies if it doesn't feed. So the second wave of neurogenesis in spinal cord will enable the slow swimming circuit to be put in place so that the animal can now start to explore with a slow forward and slow turn that I've shown you in the previous slides. And so now if we look at the escape itself, what we see is that in the larval stage, an escape will be actually a series of oscillation, like I said, with high amplitude, high frequency, going lower amplitude, lower frequency here. And in the tail angle over time, what you can see is that you have a section of the escape that will occur at above 30 hertz, so where the primary motor neurons are going to be recruited. And then another section that would be what we call the slow component, where the secondary motor neuron that are also the one recruited for slow swimming during exploration are recruited. And so if we go now to the question of the sensory feedback, in this animal, you have different kind of sensory cells. So you have lateral line hair cells that are on the skin of the fish. They are grouped into neuromasts, and they are sensory cells that look very much like hair cells in the ear that can detect mechanosensory inputs. I see that Priyan has a question. I have a small curious question about the design of the escape experiment. As far as I could understand, it doesn't seem like the presence of the prey was transient and was persistent for a short time. Okay, so yes, so I think there was a little confusion I need to resolve. So the prey capture is occurring when the fish is in control and is trying to hunt. So I'll just show you that one again. Yes, here. So in this case, the animal is hunting for the prey and is the predator, okay? In the other case, like this one, the larva is actually the victim of this predator, the dragonfly larva, and it escaped by doing this very large C-band. So a small stimulus moving and looking like, a prey will elicit the prey capture response that you see here, with a series of bouts that we're going to discuss more tomorrow. And then the escape will occur when the animal like bump into each other, that can be one, or when you scare them with an aversive stimulus. For instance, we can use acousto-vestibular stimulus, where we use just a sound, very high-pitched sound, and we make, the speaker delivering the sound is coupled to to the plate where the fish are staying. And by having those vibrations occurring at high frequency, you can scare immediately the larva and elicit an escape response, in which case it will be, very fast and high amplitude movement, different from what you get when they are capturing prey, and they tend to do small movement to try to align themselves and then eventually capture, swim faster and forward to get the prey. Okay, so this is the escape response. So really a big predator going after you when you're trying to escape, or a sound, a loud sound, or aversive chemical cues can elicit a large escape response. So we use typically in the lab the sound because it enables you to be very good at controlling. It's very stereotypical and it's very, it's clearly massive and fast, the response you get for the escape. Now, if we ask, okay, how does the sensory cell feedback onto this, motor circuit in order to possibly modulate locomotion? We have different sensory cells. The lateral line hair cells are little hair cells similar to what we have in the ear, except that instead of detecting, sound, they will be localized on the skin, forming this, neuromast structure here and detecting change of the flow. So like if the fish is feeling like, again, a big predator is coming, there might be a change of flow associated with this big predator, and they can detect it via the lateral line. Then we have neurons that will be detecting response to touch, also to nociception. So we have in green the round beard neurons that are big cells present in the spinal cord. You can see them here, together with the dorsal root ganglia neuron that I've shown you for the typical mammalian system, but it's also present in fish, and they are outside of the spinal cord here on the lateral view. And then there is a cell type that we've been, focusing on because it was not described in the textbook. It's a cell type that you can see here with a very tiny apical extension of ciliated neurons that are in the spinal cord. And they're precisely detecting the curvature of the spinal cord. They are present in the fish, but also in the mouse and in the macaque. So we can use this approach with genetics and optics in the fish to look now at the activity of these cells in vivo and to look at what happens when we silence this cell in vivo. So here I will show you one type of experiments we've done where we use another sensor that is called GFP echorin that I will explain to you very, very soon, that enable you to look again at calcium entering into the cell, with the advantage that you don't need to use a laser to activate, to get the probe to be fluorescent. It will spontaneously emit photon. And so it's a very nice bioluminescent probe that enable you to just collect the photon emitted by neurons when they are activated and the calcium rise. And then the other approach that we will also talk about is using now a toxin called botulinum toxin. So the commercial name for this toxin is Botox. It's what people use sometimes when they age and they want to get rid of their wrinkle. And so they will use the Botox injection to forbid the nerve ending to contract and to get the muscle to contract to form the wrinkles on the face. And so we use the same trick, but we use it in our experiments in order to now silence the output of neurons that we are interested by. And this was a transgenic line developed by Koichi Kawakami in Japan. And so then we can ask, okay, how does the sensory feedback impact the recruitment of motor neurons? Contribute therefore to the active locomotion, and how are these effects mediated in the spinal cord in the circuit that we've talked about? Sanjay?
Yeah, I had a question, and I think it relates to the last few slides.
Yes.
Which is that, larval zebrafish are quite small, so they are in very low Reynolds numbers. And then, as they move maybe from routine turns into these fast C starts or escape responses, they span maybe 1 to 2 orders of magnitudes of Reynolds numbers, maybe 1 order of magnitude, maybe going from 10 to about 100, in which then the physics of the locomotion is quite different. There's at higher Reynolds numbers, there's much more role for inertia to play, whereas at the lower ones, they'll really have to do everything with their tail. So does that factor into any of this?
Yes, very much, in fact. So, in fact, at early stage, indeed, they have, like you said, they need to do everything. So what happened is that when they initiate the active contraction of the muscle and they beat the tail, they have literally almost no gliding. So at the early stage, when they start swimming, they use 10% of their time to contract muscles to initiate the swimming.
Right.
And when they're done, when the tail angle stop, there is very little gliding, very little inertia. When you look at them in juvenile, it's a completely different story where they just need to, they've grown up, like, they went from 3-4 millimeter to now typically, let's say 11 millimeter when we're looking at them at 1 month. And then, indeed, the higher Reynolds number will lead to this very strong inertia that enable them to do a very few tail beats and then glide for a very long time, which actually led scientists to think that they were not doing beat and glide anymore because they looked like they were continuously moving. But in fact, if you look at them, we've started to do that in the lab. If you look at them at this late stage, you actually realize that they still locomotive bouts. They're just not continuously contracting the muscle, and they're taking advantage of the inertia to move for a long time after they stop beating the tail. And that's actually something that you're going to see with the approach that was designed by Gautam Reddy tomorrow. We can take advantage of, because by having this interest into where does the movement come from, where is it generated, we distinguish the active contraction from the passive movement. And it simplifies the problem a lot then, because then you can identify with clustering the different bout type every time the tail is contracting actively. And then establish whether, in the example you're going to see tomorrow, there are like 7 bout types that we use and call like letters. And then we can ask ourselves, okay, so how are the letters put together in order to form a word? And that's a question that we ask looking at different kind of behavior. Yes, Sanjay?
Yeah, and so a second part of this is that, of course, what they're sensing with their lateral line has also a similar effect. That's Reynolds number dependent. The boundary layer would be much
on
greater.
that one, I don't think it's been investigated at the late stage to my knowledge, but what people have found is that while the animal is moving, there are in fact neuromodulatory pathways that are able to shut down the lateral line when the animal is moving.
Ah, nice.
So the animal needs to distinguish what comes from itself compared to what comes from a big predator, right? So they're basically not sensing through lateral line during movements, and they're using only the downtime to sense.
That's interesting.
Yeah. And it's actually really cool because it's not the case for all fish. So if you look at the cave fish from Mexico, when they are raised, after many generations, cave fish that were Astiniax mexicanus that are in the cave for many generations, they're still able to breed with their, counterpart from the non-cave. So they are the one that are outside in the light, but they lost their eyes. So they destroy their eyes, and they are basically like blind fish. And together with blinding, losing their eyes because they don't need them in the cave, they also adjusted their lateral line so that now their lateral line is so critical because they don't have vision to help finding a predator, right? That they're using it continuously. And so in this animal, for instance, the lateral line is never shut off and it's always active, so that even when the animal is swimming, it's detecting that there might be a predator. And in that case, they use differently the information of the corollary pathway, saying that the fish is swimming because they're continuously relying on it to detect predators.
Yeah, I mean, the lateral line is also much more morphologically different. There's a lot more superficial neuromass all over their face.
Yeah, yeah.
which is,
They've really changed the morphology of the sensory system.
yeah.
For sure. So what is interesting is to think, okay, we clearly are not looking at humans here. We're looking at a little fish, but what could be the effect of the sensory feedback in this fish? How do we see it using optical methods for reporting the activity with and without feedback? And then using the botulinum toxin light chain I've shown you to silence the output of neurons. So we can silence the output of different kind of feedback, the excitatory glutamatergic sensory cell or the inhibitory GABAergic sensory cell. And then we see what happens to the behavior. Okay. So what would be your prediction? Does anyone want to make a bet? What would be the effect of removing the feedback? At this stage, you don't know, so you can really say anything. Anything?
I think they're shy.
Okay. It's okay. It's okay. So if you get, I'm going to go there only in step 2. So if you have an idea, don't feel bad. Just raise your hand, and I'm happy to hear it. So we're going to go through the first part, which is like, how do we look at the activity of neurons? With this problem that we have movements during the during locomotion by definition. So how do we look at it? So one path that we took that I think is worth sharing is the fact that we can use bioluminescence to collect activity of cells without having a laser to activate the sensor, and just collecting all the photons that are emitted when the cells are active and exhibit a rise in calcium. And so I'm just seeing that there is a comment here. Yes, so Sanjay, you're right. They will still generate rhythmic tail motion. So indeed, like, Stan Grillner and others have promoted the idea. In the 80s, the central pattern generator will not need the sensory feedback. So they will still be able to swim in some sense quite fine. And we'll see, it's more in the finesse of the modulation of their swimming that there are differences. And I'll be curious if you have any ideas or bet or intuition, or just, want to play, let me know. It's not coming right away. So we use 2 approaches. One is to use a regular calcium sensor that I've shown you for GCamp, and the other one, so it's a fluorescent calcium sensor. And then the other one is the bioluminescent sensor that I mentioned before. And so here I'm showing you one experiment that we did with my grad student Urs Bohm, where we looked at those sensory cells that I mentioned to you, like ciliated sensory neurons inside the spinal cord of vertebrate species. And they are indeed GABAergic neurons. They are all along the spinal cord. And Urs looked at animals in which those cells are expressing GCamp. And in order to find out what the cells were doing, he had to fight one problem, which is that if the cell is going outside of the focal plane in the imaging, it will look like dimmer, even though it didn't change maybe brightness. And on the contrary, if the cell was slightly off the focal plane and is going back into the focal plane during the recording, it will look like more fluorescent, meaning like calcium increase, the cell is activated, when in fact it's just an artifact of the motion itself from the movement. So in order to solve that problem, Urs used an approach that is quite interesting. It might be useful for you if you want to do experiments when the animal is moving as well in different kind of species. You're combining the GCamp activity sensor, so it's really a calcium sensor, fluorescent, together with another fluorescent protein, in this case, a red fluorescent protein. And it's by doing the ratio of the activity sensor to the position sensor that you can actually detect what is the true response activity of the cell. Because when the cell goes out of the focal plane, you will lose your signal both in the green and red, and therefore you can tell it's an artifact. And by doing this normalization, you can monitor what happened in the fish when it's bending its tail. And the way we elicit the bending is by puffing in the otic vesicle on one side, so that we can get the animal to respond with an escape when it's bending, the tail on the other side. And of course, I told you it's moving really fast during an escape. So we're using a little trick that, Vatsala is using as well for our experiments, where you put the fish in agar so that the head is stuck and you don't have any, you don't have a real escape, you just have the movement of the tail. And we need to follow the behavior at high speed. So I'll just check the question before I move on. So they will not deviate, correct, alter their motion. I guess they may not be able to adapt and perform fine movement without sensory feedback. Perhaps their ability to perform pre-capture is affected. So that's really interesting. So you're going to see, so indeed they do keep, like you're right, they do keep their basic oscillations the same. And what they change is much more the fine, refined movement. And then we will see exactly in which direction that goes. So first, we're looking at the activity pattern. So we're using a 2-photon microscope where you have a very thin optical slice. When the animal beats the tail, you are losing, the cell transiently from the plane. It's happening here. And then it comes back into the plane, which is the beauty of it because the fish has the head fixed. So it goes back into the plane. And after a few frames where we cannot detect the cell anymore, it comes back into the plane and we can detect the fluorescence of the GCAM, the fluorescence of the red sensor. You see there was a little shift. The cell is a little bit more in the plane than it was before. And then we can correct by doing the ratio and looking at the variation of the ratio in the experiment. And by doing that and monitoring what happens during each tail bend, we're comparing left and right tail bend. What also has been able to show is that these sensory cells that are, all along the spinal cord, organized in left, right, and ventral cells, are in fact able to detect the curvature of the spinal cord in an asymmetric manner, so that when the curvature occurs on the side, so that we have a concave bend on the side of the contraction, we're activating the cells that are immediately located on that side. And this is not the case for the cells that are on the opposite side. And so that was the very first experiment in 2016. And since that, we've shown that the ventral one are activated when the animal does longitudinal contraction, so the equivalent of crunches for us. And that indeed this sensory system is able to detect left, right, and ventral by interacting in the central canal with a very long polymer that enable the activation of the apical extension that they are bearing here. And so this is one way to do it. And we confirmed that it was the case by looking at the change of flow. We're using fluorescent bead in the cerebrospinal fluid. And we find that every time the animal contract muscle, they are flushing the fluid really fast. And that corresponds to a very strong activation of the CSF contacting neurons. Now we used another method that enables to look at activity of cells when you don't want to use a laser because the movement is going to be too large and there is no way to compensate for it, like the way I showed you for small movement with red and a green sensor of position and activity. And so another way to do it is to target a sensor for activity only in the cell type of interest and use bioluminescence instead, where the photons are going to be emitted by the neuron during activity. And we're collecting all the photons emitted in order to know when are the cells active. In this case, because there are very few photons expressed by the cell, we can't really do the same kind of imaging. We don't have enough photon for an image. And so what we do is that we just collect the photon and look at one single signal, the number of photons emitted in the entire animal. So it's very different experiment. And here is how it looks. So it was done, it was used by Eva Norman in a paper where she did the proof of concept. And in our case, we used it to see, well, what changes in motor neuron activity with and without the feedback if we paralyze the animal or if we look at mutants that don't have muscle contraction. So we put the little larva here in the dish, and then we have a camera monitoring its movement. It's freely swimming, and we're illuminating the animal with an infrared LED so that we can see the position of the fish. And then underneath, we're really using a photo multiplicator tube to actually count all the photons that are emitted by the larva. And we're collecting all the photons that we can collect from that side in order to see, well, what happens to the recruitment of the motor neurons. And so what we've spent a lot of time with Stephen Knafo to optimize this experiment is to actually make sure that in the fish, there was no muscle fiber expressing the echorin. Because you can see here in those animals that we have very nice expression in cells in the spinal cord. And you can guess from the projection outside of the spinal cord that are going on the ventral musculature that we're labeling the motor neurons. If you look at a close-up, you can see very nicely the motor neurons are there. They're forming a very nice pool with the axon going up and down into the muscle. And we need to be sure that we don't have a muscle fiber, because if there is one, it would lead to a gigantic calcium response and it would really mess up our experiment. So we've been really careful at generating this animal and checking that there was no muscle expression. And then we got really excited because we could manage to get, after an acousto-vestibular stimulus, a response of the fish where they're performing this escape. We're simplifying our problem by keeping the animal head in agar again, and we can monitor the tail angle over time while collecting photons. And you can see the scale is 4 photons. So you see that we're collecting about 16 photons. So there's not that many photons that we have, but we can collect signal just from motor neurons. And before it was used by Eva for pan-neurons, so with many neurons. So when we started those experiments with Stephen, we've looked at different response of the fish, some in like the case I showed you before, where you have a full C-bend and succession swimming. So it looks like an escape response. Some where you just have a C-bend, like you can see here. And some where you just get, in fact, a slow swimming, so you don't get real escape. And so we can see how the signal in the motor neuron change. And what we find is that for, typical escape response, we have quite large, response of the bioluminescence calcium sensor. For the C-band only that are much larger amplitude, typically, we get an extremely large response showing a large recruitment of motor neurons. And for the slow swimming with small amplitude of the tail bend oscillation, we get a small response, small signal from the bioluminescence. So we have a good readout of how much the contraction occur. And for the largest amplitude, we get the largest signal. Even though, of course, there is quite some noise, because in order to get the bioluminescence sensor to work, you need to feed the animal with the selanterazine. That will be a cofactor that is necessary for the emission of photon. And this process is going to be different slightly from one animal to the other. And therefore, you get some more variability than what you would get with a typical experiment using just a genetic encoded sensor like GCamp. So if we compare what we get now with GCamp in a fictive preparation when the animal is paralyzed and we're recording from the ventral nerve roots doing the exact same experiment, what we can see is that during the slow swimming, we get the small secondary motor neuron active. And when the escape occurs and the swimming occurs above 30 Hertz, then you start getting those very large primary motor neuron, which is also what the group of Fetcho had described with electrophysiology. And so now what is nice is that you can see, well, what happens if you try to block the contraction of the muscle and the recruitment, therefore, of the sensory neurons. And what we've compared is, a freely swimming, a head restrained swimming fish to an animal that is either paralyzed or has a mutation that makes it unable to contract. And in both cases, we saw the same effect on the bioluminescent signal. when you compare active animal to animal, the same animal that were paralyzed with pancuranium bromide so that they can't contract the muscle anymore, you see a reduction of the amplitude of the bioluminescent signal, meaning that you have less activity in motor neurons overall when the sensory feedback is removed. And this we also get when we're comparing now control siblings in blue that are motile with homozygote mutant for a mutation that will lead to paralysis of the muscle. And you see that you also have a reduction of the amplitude of the signal when you compare the siblings, everything being the same. So now what could be the cause and what would be the readout in terms of behavior of this reduction in the recruitment of motor neurons? Is it a change in speed? Or is it maybe a change in the amplitude of the movement when the animal is trying to move? So we looked at that by using the tool that I mentioned to you, this botulinum toxin light chain that enable you to block the release of neurons. And so what is nice with this toxin is that it's genetically encoded. It's just the light chain, version. It's very small. So it's easy to have transgenics expressing this toxin fused to GFP. And then it acts on the VAMP and block the synapto. It's also called synaptobrevin here. So it blocks the ability of this snare to fuse the vesicle to the membrane, and therefore there is no more release at the presynaptic site. So we showed with electrophysiology that on a connection that we had identified from the sensory neuron I mentioned that contacts the cerebrospinal fluid to the motor neuron, we have a typical IPS-C that we can record here that is blocked by gabazine. So it's a GABAergic current. And then if we look now at the animal in which the opsin is co-expressed with the botulinum toxin, so that every time we activate the presynaptic cell, we're also blocking the release, we don't have any more a response in this primary motor neuron that we always find in the control. So this method has been very useful to actually check which neuron does what. And as expected, if you try to stimulate with acousto-vestibular stimulus, you're getting this nice escape response. And now if you're using a line to block the release of motor neurons so that they can't talk anymore with the muscle fiber, you don't have any motion. So you have a complete paralysis. The tool is very effective. If you use it with a line that blocks the output of all glutamatergic cells expressing VGLUT2, a transporter that is mainly used in motor circuits, you also get full paralysis. And now if you try to target it to the V2A cells that I've shown you are important in the central pattern generator, you don't fully block, but you reduce a lot movement. And that is also shown here, where for the escape response, animals where the V2A neurons are silent will still do a tail bend, but it will be lower amplitudes, and it will just be a single tail bend. And that is due to the large Mosner cell that is a common neuron that drives the escape that is still functionally active in these animals. So you have a clear difference of the kinematics, and you just get single tail bend, lower amplitude. So now we've done these experiments, and I want to point out for anyone who is interested in behavior that the behavior experiments are very fast, but they have a limit, let's say a feature that is very interesting at the same time. You need it's a challenge, is that it's very reliable and stereotypical, the behavior I've shown you for the escape response, but it's also very variable, so that if you're looking at every dot here is one fish for which we elicited multiple stimuli. So we use multiple trials to look at the response, and you see that there is a huge variability going from almost 40 millimeters per second in speed all the way to 100 millimeters per second. So that means that if you have such variability, you actually cannot really look at 3 animals in order to conclude. You need to really look at many, many animals in order to be sure that your effect is there from one clutch to another. It's also there from one day of experiment to another. And it's also there from, one lab to another. So you need to really like establish that your behavior is steady by indeed having large ends to go around this problem. I see that maybe you want to remind me that there are 10 minutes left.
That too. One
Okay.
quick question. So the
Yes.
Botox construct, does it induce any developmental defects having this in the animal when it's growing up?
Yeah, so this is part of the story that by student Jenna, we were so hopeful that there would be a developmental defect. So Jenna has spent a lot of her time to actually look at that. And it was, sadly not the case. So she didn't get, she tried it on GABAergic neurons and glutamatergic neurons. And on the CSF contacting neurons as well. And when she looked at those animals and quantified the population in those in the spinal cord, she didn't see any effect that was very striking. So we don't think that there is huge effect of the activity during development. In fact, maybe there are some. But
So
you
it
would need to do electrophysiology.
Does it shut down spontaneous release also, or does it shut down all transmission?
That's a good one. In this particular experiment, we actually recorded only from the motor neuron. And so you see that, of course, there is still spontaneous activity from other premotor cells, but we didn't really look at whether you would still have GABAergic minis. I believe that VAMP is actually necessary for that as well. So in theory, at least, but we didn't demonstrate it, you should also block the spontaneous release.
Okay. Yeah, thanks.
But to be honest, very fairly to the massive effort of Jenna, she only looked at morphology and number of cells. So it could be that you have physiological changes. And I think it would be a very good tool to tackle this question, because like we did, we really block all release on the evoked release. Because VAMP is such an essential snare, it should also be necessary for the spontaneous release. So I think it's really interesting to dig into that one. Okay, so now having 10 minutes, I did like half of what I wanted to talk to you about. But what I want to insist is on the fact that, and I will just cut it short to make it a story complete. When you try to silence the feedback from glutamatergic sensory cells, so in this case, we go for both the trigeminal neurons, the round beard neurons, and the dorsal root ganglia neurons. So it's quite a large population of sensory cells. We're getting this effect where we have a reduction in speed that is associated with a reduction in the tail bit frequency. The effect you see is typically of 20%. So it's a small but very reliable effect. Of course, there might be compensation because the Botox is going to block release all along during development. So it's possible that there are compensatory mechanisms that are going on. In the case of the GABAergic CSF contacting neurons, we're getting a smaller even effect. We're talking 100 cells only at most. And in fact, half of them don't express the botulinum toxin. So it would be 50 cell silent. And you can get a reduction again of speed and reduction of tail bit frequency when you silence the sensory feedback. And so what we've done then, I really want to illustrate that point, is that if you start from the behavior and then you try to dig into it as much as you can, you find the solution of what could be the modulation at the circuit level. And so what we've seen in the escape response of the animal, where in this case, it's glutamatergic sensory feedback that is silenced by Botox, we didn't have any difference of the distance travel. We didn't have any difference on the number of bends that occur during an escape, which is typically 6 on average. So 6 oscillations. But what we found is that there was a very interesting increase in the duration of the escape. That was, as a matter of fact, because you don't change distance, associated with a reduction of speed, like you can see here. And the reduction of speed is corresponding to a reduction of the tail bit frequency. So those are all the average value of the tail bit frequency and the speed. And then we went back and said, okay, now let's look at the fast and the slow component, because we know that in the spinal cord, they are handled by different motor neurons and also by different premotor interneurons from this V2A family I mentioned before. And so there, it was really interesting what we did when we compared the fast and the slow, by this color code that you've seen before. We find that most of the modulation, in fact, is occurring in the fast, where you can see that the duration of the fast swimming will be reduced when you silence the excitatory mechanosensory feedback. Also, the number of bends will be reduced that occur in the fast, and the tail bit frequency will be massively affected when the fish is in that fast regime. And what is interesting is that the total number of bends is not affected, because as a compensation, you have more oscillation occurring in the slow regime. And that's how you keep your number of oscillation constant, even though you have fewer oscillation in the fast and more in the slow. And so we thought like, that's really neat, because some prediction of experiments in invertebrates this time were really predicting that the sensory feedback was critical for optimizing timing, and could optimize timing, or could optimize the amplitude of the oscillation. Now, if we look at what happened in the very first experiment, where they compare the fictive and the active locomotion of a lamprey in 1984 by Thelma Williams and Patrick Whelan, what you see is that there's something that they didn't mention in the paper that is striking by eye. If you compare those two, you're recording from the muscle here in a freely swimming lamprey, and you're recording from a lamprey dissected in a dish after putting, in this case, glutamate in the dish. What do you see differs between this recording and this recording? Looks very much the same. You have bursts of activity alternating left and right, and going from head to tail with segments that are low, like 5, and then going all the way to 47. But what is different, do you think? They didn't see it in the paper, like they didn't mention it in the paper. I truly believe that they didn't really notice it as well. But do you find a difference?
There's some responses in the chat box.
Okay, cool. Burst duration. Yes, thank you, Arnap. It's exactly that. So it's exactly that. You have the scale bar here of the time, and you see that in the intact animal, a cycle lasts typically around 100 milliseconds, 0.1 seconds, while in the fictive, when you dissected everything out and you don't have muscle and feedback, and it goes 10 times slower. And what is remarkable is that if you go to SFN and you look at all the poster comparing, what's going on in the fictive to what we know is that going on in the active locomotion, similarly for mouse, for cats, for everything, you see the same effect. In fact, it's a massive difference, which might explain why robots are much slower than what we can get with animals that they don't have the feedback implemented like we do in the biological system. And in fact, this feedback is really interesting. That's an experiment from Greener himself in the 81, where he used the tail bending of the lamprey in order to entrain the CPG, and he could get the oscillation to go by just mechanically entraining the tail. And so the feedback is really necessary to speed up, and it's even more necessary if you have limbs, but it's very true for aquatic animals. Another thing, and I will finish on that, is that the feedback is also necessary for maintaining posture. So this one is now looking at the escape of animals that express botulinum toxin in the CSF contact neurons. We saw a mild effect on speed and tail bit frequency, but when we looked closer, we saw that there was a really interesting effect on posture. And the animal without this feedback to tell how curved the spine is in 3 dimensions, left, right, and ventral, loses the ability to maintain its position and start rolling in 3D, like it was mentioned by someone in the beginning. And so we developed deep learning algorithm to analyze this, in real time and find out that indeed, without, just with ablation of 60 of these cells that are projecting into the hindbrain, we can affect the rolling of the animal during the escape response. So I don't have time to present you how from this behavior we dissected the circuits. I will see with the 2 GOTAMs if I can take 10 minutes maybe to explain based on this observation how we understood with optogenetics how the circuit was wired. I can probably just take 5 or 10 minutes of the time tomorrow to continue on that. And then with them, you will have an occasion to start thinking about another, level of understanding of movement, which is like, how do we form now sequences of action? Okay, so I don't want to make you late. It's exactly, I believe, 2 p.m. for me, so it must be 6:30 for you. Is that correct? If that's okay, I will finish with 10 minutes tomorrow before you go into the modeling part. Okay?
We can take a few questions.
Okay, great.
So while people are, preparing to ask their question, maybe I'll start off. So one thing that strikes me when I look at this drawing of the CSF neuron is its process is going into the central canal, right? Mm-hmm. Do you think there is tuning either in terms of frequency or direction of motion for these different CSF neurons that may, so when you silenced the transmission from all of them, right?
Yeah, right. We silenced either the transmission from all of them, or in the experiment of Mingui, we ablated the 60 rostral modes that go to the brain. And we didn't distinguish left and right. But it's, so what we see is that there is such an asymmetry in their detection. They can detect left or right bending. And they project only to one side. So they project only to the ipsilateral side. So what we expect indeed is that if you just ablated the left one, you should see a defect, in the control of the motor neuron on the left side only. And it's an interesting experiment that we didn't do yet, actually, to try to make an ablation asymmetric and see the effect on the behavior. Yeah.
So where I was going with that actually clarifies a lot, but where I was going with that was along the rostrocaudal axis, could there be different subpopulations of these neurons such that, for example, the tip of the tail is typically involved in more slow swimming versus the mid-body might be involved in fast escape.
Yes, totally. No, that's really fascinating. The work of Moini, who was your former grad student, is showing very nice patterning of the connectivity along the spinal cord, which is revising a lot the way we understand the CPG, right? It's not the same unit, it's connected differently, different location. And we have evidence that these cells have different targets when they are the rostral-most in the brain. They don't project onto the same cell that when they're in spinal cord. But we didn't trace the connectivity along the rostral axis in the spinal cord. So we clearly think, like we saw for the curvature plot, that you have differential activation during the locomotion. And we see clearly that the way we ablate them will affect or not the behavior. So if we ablate the rostral most, we affect the C-bend of the escape, but we don't affect at all the slow swimming. So that's clearly there, but we haven't done asymmetric manipulation. Yeah.
Okay. Thank you.
Thank you so much. It was really nice. If that's okay with you, I will end it here and then continue with just, maximum 10 minutes tomorrow, where I want to show you the circuit analysis that we've done with optogenetics based on the behavior phenotype. So both for the rolling and for the speed modulation. And then I will leave the floor to the 2 GOTAMs, GOTAM Ready and GOTAM Sridhar, to show you what they've done more analytical this time on the analysis and segmentation of sequences and the behavior. Is that okay for everyone?
Sounds wonderful to me.
Okay, thank you so much. Thank you for your questions. See you tomorrow. Bye-bye.
Thanks. See you. Bye.